Hello everyone, welcome everyone. Today I will teach you an introduction is that referred to as a transportation of crop plant genotype from one place from their original or cultivation to such an area where they have not been grown before. For example, we plantation of potato in India in Bas, so we take it from the anole, and some other examples are also there. And plant introduction is the one of the oldest and effective method of plant breeding. The main function of the plant introduction is to make available the germplasm that can be utilized in the plant breeding in program. And there are two types of plant introduction are there. Number one is plant introduction. Plant introduction means is introduction without any alteration in their original genotype. In most of the commercial flower crops and rose, primary introduction is widely adopted in the techniques of increasing the gene pool, and the secondary introduction. And in the secondary introduction, the variety is subjected to selection and to isolate the superior variety to make hybridization with a local variety and the transfer with a more character from the local variety. And next is what is the purpose of the plant introduction? What is the purpose? The main goal or purpose is like that to obtain what entire new crops and to serve a varieties as a new variety and to use in the crop improvement. And we can use the crop improvement, for example, rice resistant varieties is also there, wheat resistance is also there. We can borrow from others and to save the crop disease and pest and the scientific study the aesthetic balance. And next one is plant introduction agencies of India. In India, the RARI in New Delhi in 1946, the introduction of plant agency. And these agencies began the plant introduction is division of botany and funded by ICAR. In 1961, the independent division is subsequently known as the National Bureau of Plant Genetic Research in 1976. The NPGR is now they are established in the New Delhi and the responsible for introduction plant agriculture and horticulture crops. And FRI, the Forest Research Institute Dehradun is in the Dehradun, and for their responsible is for introduction of forest trees and vines. And the Botanical Survey of India is also there, and they are the agencies of India. And there are 5 procedure for the plant introduction. Number 1 is procurement, number 2 is quarantine, number 3 is cataloging, number 4 is evaluation, and after that the last one is acclimatization, and number 5 is multiplication. Number 6 is multiplication. So let's discuss a little bit and about the procurement. The new germplasm in procedure through the NPGR is New Delhi, and after that this one is generally material is obtained from the corresponding or as a gift or exchange germplasm in a considered fast gift euro and the anticipation of future. And the second one is just like quarantine. When we do our COVID, we do take some quarantine like 15 days in home without going outside and all, just like that. And we should keep the plant as a quarantine. This process, like the plant are suspected and contaminated, fumigated, and given isolation for the treatment, observe insect, pest and disease. This entire process is known as a quarantine. The rule which are used for the quarantine are known as quarantine rule. Yeah, this one is quarantine rule, and the quarantine rule should be proceeded to 21 days. It should be 21 days. And cataloging is that like all the plant material which are introduced in the given entry number, information regarding agencies, places of the origin, adaptation means where they are, which plant, this plant is given by some agencies, name, tag regarding, and where that is the origin, and what is the adaptation of the plant. And in the plant material classification, like exotic collection, well, we have collection, and exotic collection, indigenous collection, indigenous wild collection is also is there. And the evaluation after that collection and all, we need to evaluate. For example, the plant material are sent into the substation bureau and evaluated with a respective to the various characteristics to assess potential of new introduction. And after evaluation, we need to do acclimatization, is the process of adaptation the new varieties to the environment. And the cross pollination lead to further gene recombination and the sub pollination. This one is this. And after that, at last, multiplication. The plant material which is introduced to be multiplied and further tested in the various location. After that, we'll need to do multiplication and we'll test it in the various location. And this one is when we do multiplication, the stability of the cultivation in different region of the country should be assessed before using a commercial variety. Why advantages of the plant introduction which give entire new crops and which has not been grown earlier. For example, bird of paradise, and it is also provide the superior variety directly or after the selection and hybridization. And many superior varieties are the flourish culture, different has been rose in introduction in the country, the direct cultivation, for example, passion, gran, gala in roses. This one gala, passion fruit, this one. And introduction and export are the only feasible and the fastest means of collecting germplasm. Collecting germplasm for in quiz and all, also for example in the higher examination like that, they can ask what is the fastest collecting germplasm, what is the fastest means. We should answer like plant introduction is the fastest means of collecting germplasm. And the procedure of plant introduction and it is the quickest and economical method of crop improvement, particular when the introduced or released varieties are directly after sample, and if the particular location is prone to some particular disease or the insect attack by the plant may introduce in a new disease free areas are directly to protect from the damage. Thank you guys for watching this video, and if you like this video, comment down below. Thank you.